All right, happy Friday, everybody. So, a couple quick reminders as we go through here. We're going to lecture today, lecture on Monday, and then we're going to have the Mythbuster oral presentations on lab class on Tuesday. We will be starting those bright and early, right at 8 a.m. Just simply because of the fact that we have so many presentations to get through. I think there is a pretty good chance we're not going to get through all the presentations. I don't want anybody to feel rushed. I don't want to try to squeeze everything. So there is a chance that if, and again, I'm going to have you guys draw numbers out of a hat, basically, to figure out the order. But there's a chance if you're one of the later presenters, you might be presenting the Tuesday after fall break then. So we would put the presentations as part of the following lab class. Okay. Fall break then is next week. So also a reminder, we don't have class on Wednesday, Thursday, or Friday next week. With that all being said, when we come back afterwards, I'll send out an email reminder about these, but we do have a lab quiz when we come back after fall break. Exam 2 is going to come up on the second Tuesday back from fall break on October 22nd. Then. So just be aware that some of that stuff's going to come back, and I'll let you know when we get to it. And no, we're not to it yet. Well, I'll let you know when we get to the break in exam 2 content. Okay. All right. Questions about any of that? All right, so today then, what we're going to do is kind of finish up talking a little bit about control of microbial growth. And that's going to lead into our next topic, which is about nutrition and metabolism then as well. So we had last lecture, we talked about some different environmental parameters we can change and control microbial growth. We talked about things like osmotic conditions, oxygen concentrations, pH, radiation. Another environmental parameter that can affect microbial growth is pressure. And so, for instance, some organisms kind of live at the normal 1 atmospheres of pressure. That is typically what we consider the environmental pressure at sea level. So at sea level, we might have 1 atmospheres of pressure, for instance. And typically when we're talking about pressurized environments out on Earth, what might be a pressurized environment out on Earth? A high pressure environment. Yep, deep sea. So typically when we're talking about high pressure environments, though, we might be talking about 1 atmospheres at sea level, but as we go down in water columns, especially as we go down deeper and deeper and deeper into the ocean, then we get increasing pressure. Anybody read up on a whole OceanGate submersible Titanic thing in the news? That was in the news a year ago. It's been in the news again recently because there's like congressional hearings on it. But first off, anything that ends in gate is always a scandal, and they named the company OceanGate. But anyways, what would end up happening here is as you go down, unfortunately, we saw that failure of the submersible, because as you go down deeper, there's more pressure, pressing. And that is actually any types of organisms that are living in that water column that are subject to that pressure. So as you go deeper and deeper and deeper, so this might be depth of the ocean. And as you go down that, so for instance, as you get down to say depths of 10 or 11,000 meters under the surface of the ocean, you're looking sometimes at over 1,000 atmospheres of pressure involved. Versus, again, air at sea level is 1 atmosphere of pressure. And so any organisms that can live under high pressure are barophiles. So barophiles are any organisms that can live in high pressure environments. So typically any kind of things that you might see living down here at the bottom of the ocean are typically going to be barophilic organisms. It could be bacteria. It can be those like little anglerfish from like the Finding Nemo movie that have the lights that attract their prey, for instance. But these are going to be barophilic organisms. Barophilic organisms have special adaptations to survive under high pressure environments. And some of them, we don't even know what they are in some cases. Actually, one of the main problems with pressure in non-barophiles, in organisms that are not barophilic, at high pressure, it causes DNA replication to fail. And we don't know why, actually. So you take an organism that's used to living at 1 atmosphere pressure, you put it under higher pressure, DNA replication doesn't work properly inside of its cells. Cells can't go through DNA replication, can't grow, can't divide, for instance. Then. And they don't know why. Barophilic organisms have some way to get around this. And again, we don't know why it fails under high pressure. We don't know how barophilic organisms solve that problem. And so it's unclear why that is, but they have to have some type of way to get around that issue. Questions about that? But we use pressure sometimes to things like, for instance, in autoclaves. Again, autoclaves, we can use this to kill microorganisms, sterilize equipment, sterilize media. And autoclaves do a combination of both very high temp and very high pressure. They actually go up to about 15 to 20 atmospheres of pressure. And that by itself is actually enough to kill most things, except for barophiles. Now, the other thing we commonly use to kill microbes or then to limit their growth is various different kinds of chemicals. And there's a lot of different chemicals we use to control microbial growth. So I'm just going to go through some examples very quickly. I could spend entire lectures on this and modes of action. For instance, some types of chemicals are known as protein denaturants. What does it mean when I denature a protein? Yep. So protein denaturants, these are things that unfold proteins. Alcohols like ethanol or isopropanol, which is rubbing alcohol. You guys might have even used rubbing alcohol to like clean a cut before. Rubbing alcohol or hydrogen peroxide is commonly used to do that. But those are protein denaturants. They kill cells because at high concentrations, they cause their enzymes and proteins to unfold and the cell cannot function properly. Halogens like iodine. If you ever have a surgical procedure, one of the things they typically do is where they're going to cut in through the skin, they treat it with a halogen solution, an iodine solution. And that is a protein denaturant, a pretty strong one. And it denatures the protein so well, it often then kills the cells involved. So that sterilizes that area. Anybody go to the dentist and they got that slightly brown liquid you got to swish around your mouth now before they start everything? Do your guys' dentists do that? Okay, well, mine does ever since the pandemic. They make you do a mouthwash rinse before they start working on your teeth. At my particular dentist, they've done it ever since the pandemic, but it's actually an iodine solution. Anyways, no? Okay.
How bad does it taste?
Huh?
How bad does it taste?
Actually, it's not that bad. It's better than mouthwash. I thought about looking to see if I can buy that from the store instead. Okay. Poisons are other types of chemicals that. Poisons are things that inactivate proteins. So an example of poisons would be heavy metals like silver, mercury, or arsenic. What they do is they don't cause proteins to unfold. Instead, they affect the active site of enzymes. So they get into the active site of the enzyme and cause it to stop working in some way. And you guys, am I okay with active site? Active site is the part of the enzyme that directly touches and interacts with the reactants, the substrates then. So they don't unfold the entire protein. Instead, they inactivate the active site in some way, and then the enzyme cannot work. We have some chemicals, like you've probably used Lysol before. Lysol spray disinfectant. A lot of soaps, detergent molecules are membrane disruptors. So Lysol is a phenolic compound. It's a phenol group. So it's a benzene ring with a hydroxyl group attached then. That is actually an amphipathic molecule. Soaps and detergents are amphipathic molecules, but those kinds of molecules have a charged side and an uncharged side, and they like to insert into the phospholipid bilayer. But if enough of them insert into the phospholipid bilayer, it disrupts that phospholipid bilayer and can put holes in it. And then the cell lyses open. And that's how something like Lysol as a disinfectant works then, for instance. We talked about other types of chemicals that I don't have up here, like bleach or hydrogen peroxide. Those do oxygen damage, for instance, then. And then we have chemicals like antibiotics. Antibiotics are chemicals that we're going to talk about. I'm going to have an entire lecture just on antibiotics later in the semester. But they specifically target structures or reactions that are required for bacterial growth, but not growth of human cells. So antibiotics are chemicals that target something that is specific to the bacterial cells. Bacterial cells have it. They stop that part of the bacterial cell, but it's some part that bacteria have that human cells don't have. Can you guys think of an example of something that bacterial cells have then that human cells don't? Yep. And a common target for many antibiotics is peptidoglycan. It's not the only one, but peptidoglycan cell walls is something that bacterial cells have. They have to have them. They need them. And that's something that human cells don't. So for instance, penicillin is an antibiotic. Penicillin targets the peptidoglycan cell wall of the bacteria. That will kill the bacterial cell and it leaves human cells unharmed. Why can you not use, say, bleach to treat an infection?
First human cells too.
Yeah. Bleach is going to kill the bacterial cells or the virus is causing an infection. It's going to kill your cells just as well, though. All right. So that's a little bit on chemical inhibitors, and we'll talk about some of those more as we go through the semester. Nutrients. Nutrient availability also can control microbial growth. And different kinds of bacteria are going to require different kinds of nutrients. Different kinds of bacteria are going to get their nutrients in different ways. So what we're going to kind of shift gears about is, yeah, nutrients can control microbial growth, but we're really going to start talking about what nutrients do bacteria require, how do they get those nutrients. We're going to shift gears into that particular topic then. Okay, so nutrient, we're going to talk about bacterial nutrition. We're going to talk about also as part of this secretion and transport then. So when I say what kind of nutrients do bacteria require, that's a tricky topic. Different species of bacteria have evolved under different conditions. So we have tons and tons and tons of different species of microbes. This microbe might have evolved under cold conditions. Maybe it grows up at the top of mountaintops. Another microbe might grow in lakes. Another microbe might grow in soil. But they evolve under those conditions over time, over generations, over thousands and thousands and millions and sometimes billions of years. So those microbes have often evolved to grow best under specific conditions. And this is called niche theory. A particular bacterial species might have evolved under certain conditions, and it's evolved and required adaptations that help them be successful under those conditions. So with that being said, there's a lot of different bacteria and they live everywhere. So it kind of depends on what kind of bacteria we're talking about. Some bacteria are what are called generalists. They grow under a broad range of conditions, and they can be very, very flexible in what kind of nutrients they require. So there are some bacteria, you can put them in this one type of environment, you can put them in a different kind of environment, and they're going to be pretty much okay. These generalists will grow in a wide variety of different environments. They're very flexible in what kind of nutrients they need. Some bacteria are more specialists. These species need very specific conditions in order to survive. Some example of this might be something like a hot spring thermophilic bacteria. It can only survive in very hot, warm temperatures. Some bacteria have evolved to be very fast growers. They have very fast growth rates. They divide very quickly. Typically, bacteria that have evolved to be fast growers, this means that they have evolved and lived in what are called nutrient-rich or eutrophic environments. Eutrophic is a way of saying a nutrient-rich environment. Fast growers typically grew in they've evolved over time in these nutrient-rich environments. Some bacteria are very slow growers. Slow-growing species of bacteria, they probably evolved over time in a nutrient-poor, what's called a oligotrophic conditions. Questions about that? So, for instance, there's a lot of bacteria that live meters to sometimes miles down in the surface of Earth. A lot of times the bacteria, they're down away from the surface. Those are fairly nutrient-poor conditions. And so they tend to be they tend to be slow growers. There's some bacteria that live down deep in the soil, down deep in the surface of the Earth's crust, and they might have a doubling time measured in months to years. They might go through a cell division every couple of months or every couple of years in some cases. So with nutritional requirements, different bacteria also have different nutrient requirements. So different bacteria species often fall in some different categories. Prototrophic bacteria. Prototrophic bacteria, they synthesize everything they need from very basic precursor molecules. You can give them ammonia. You can give them some CO2. You can give them some oxygen or some hydrogen gas, for instance. You can give them very simple compounds. From those very simple compounds, they make all the different parts of the cell. They turn those simple molecules into more complex molecules like the macromolecules, the lipids, the proteins, nucleic acids, carbohydrates, where they take these simple precursors and then they put them together into more complex building molecules, building them up. These prototrophic bacteria are often robust. They're usually fairly easy to culture in the lab. Some bacteria are known as fastidious. Fastidious bacteria are possible to culture, but maybe a little difficult. Usually with fastidious bacteria, they require some or many specific compounds to be added into there. There's some very specific nutrients you need to feed them to get them to grow. Technically speaking, we're kind of fastidious. There's several different things our body needs that we cannot make. For instance, we can't make vitamin K. I think I've brought this up previously. We need it, but we can't make it. We have to get it from eating it from some type of environmental location, some type of food source, or we get it from microbes in our body is making it. That might be an example of that. And then there are many species of bacteria that we consider non-culturable. We know they're growing out in the environment. We know they grow just fine out in the environment, but we can't grow them in a lab setting. And I have 99% up here, but the estimate right now is that only about 1 to 3% of bacteria are culturable that we know of. And we don't know why. If we knew why, they would be culturable. We might be that we are missing a nutrient and we don't know what nutrient they need. Might be that something in the typical types of growth medias we use, like our nutrient broth, is as a toxin, has some type of compound in it that's toxic to these cells. It might be that they're intolerable of a certain temperature condition, oxygen concentration, salt concentration. Could be a combination of those parameters then as well. We don't know why, but we can't grow them in the lab. When we do grow them, well, you guys have been doing this in labs, so I don't think I need to go through this too much. A lot of times we grow bacteria in either liquid media or on solid media. If we grow them on solid media, we typically get the bacteria nice stationary. We can see individual colonies. We can make sure that it's not contaminated. Because we can see the colonies, we can make sure there's no contaminants on solid media. Different species have different colony morphologies. Liquid broth, can you necessarily see a contaminant very easily? No. It can be hard to see bacteria that are mixed and growing in there. It's possible sometimes, but other times it's not. But the liquid media is really nice because you can grow high cell densities in liquid media. When a cell is floating in liquid media, it's bathed by nutrients on all sides. And so what you get in liquid media is really high growth rates and a large number of cells to study. So liquid media is really good for quickly growing a large population of cells. Solid media is nice when you want to make sure things aren't contaminated or you want to store them for a longer period of time, for instance. Now, I'm dancing around the issue of what nutrients bacteria need a little bit, if you guys haven't noticed. It's because different species need different nutrients. But in general, there are certain types of chemical elements that they need. So some chemical elements are known as what are called macronutrients. What does macro mean? Big. Macronutrients are going to be different types of chemical nutrients that cells need a lot of. Okay, so macronutrients, that basically just means they need a lot. The big four are in red. They are carbon, hydrogen, oxygen, and nitrogen. 96% of a cell's mass is those four. Carbon, hydrogen, oxygen, nitrogen. They need a lot of those four chemical elements because those are the most common chemical elements in the macromolecules. So you need a lot of carbon to build macromolecules. You need a lot of hydrogen, oxygen, nitrogen. And by the macromolecules, again, I mean carbohydrate sugars, lipids, nucleic acids, and proteins, the four classes of macromolecules. Questions about that? The cell also needs then quite a bit of phosphorus and sulfur. About 1.6% of the mass of a cell is just phosphorus and sulfur then as well. Phosphorus is almost always in the form of phosphate. So I've got my phosphate here, PO4. So phosphorus is almost always in cells in the form of phosphate. And then you have a lot of phosphates in DNA, in RNA, in adenosine triphosphate, in phospholipids, for instance. And where do you find phospholipids in the cell? Membrane. Yep, membranes of the cells, then, for instance. But all of those have a lot of phosphate groups in them. Remember, DNA and RNA, the backbone of DNA and RNA is a sugar, phosphate backbone, for instance. Okay. Phospholipids, the phospho part of phospholipids is a phosphorylated lipid, in essence. Sulfur. You need a lot of sulfur for 2 of the 20 amino acids, methionine and cysteine. So methionine has amino acid has sulfur in it. Cysteine amino acids have sulfur in it. You also need quite a bit of potassium, magnesium, sodium, calcium, and iron. So these are what are considered the macronutrients, not just for bacteria, but pretty much for all cells, by the way. Almost all cells need quite a bit of these particular things. If there's macronutrients, though, there's also micronutrients. Micronutrients, they are necessary. But only needed in trace quantities or tiny. Again, trace or tiny / small quantities is really what we're looking at here then. Micronutrients include things like cobalt, copper, manganese, molybdenum, nickel, selenium, tungsten, vanadium, and zinc. What are micronutrients typically needed for, though? They're needed for enzymes and enzymes to function. They're needed as what is called enzyme cofactors. you'll see that all of these, hey, enzyme cofactor, cofactor, cofactor. Most of the micronutrients are needed as a cofactor to help enzymes function. What exactly is a cofactor? Cofactors help enzymes function. And they do it in different ways. So they could add extra chemistry to the protein. So here, for instance, I have an iron as part of the active site. And I'm going to underline active site. When something is acting as a cofactor, if it's adding extra chemistry to the proteins, to the enzymes, it's always functioning at the active site of the enzyme. Can anybody think of an example of a protein that specifically uses iron as an enzyme cofactor? Hemoglobin is then the common example given out for that. So hemoglobin is the blood carrier it carries oxygen through our bloodstream. But hemoglobin, every hemoglobin has an iron at its active site, and it's actually the iron that directly interacts with the oxygen and binds with it and carries it from our lungs to all the cells in our body. Other cofactors, their job is just help the protein fold up properly. So some enzymes don't fold up to their proper 3D shape. What's the name for the 3D shape? And what's that level of protein folding?
Tertiary.
Yeah.
Or quaternary.
It's tertiary. Yep. So some cofactors are necessary for enzymes to fold up into their proper tertiary shape, 3D shape. But also sometimes cofactors stabilize protein-protein interactions. So it might be that you have one protein, and then the cofactor helps it to bind to a second copy of the protein or to a different protein. By the way, what level of protein folding is this then basically being involved in?
That would be quaternary.
That would be quaternary then. Questions about that? By the way, metals like iron or zinc, they commonly are inorganic cofactors. You guys ever wondered why you need vitamins? Vitamins are organic molecules that then act as enzyme cofactors. So things like vitamin A, B, vitamin K, they help some of the enzymes in our body function properly.
Would it be wrong to think organic cofactors are also coenzymes?
To some degree, yes, they are coenzymes. Yes.
Okay.
And the coenzyme term is often similar in meaning to cofactors. Those two terms are kind of the same thing.
Okay.
Except they're not. Let me think about that and I'll get back to you. Okay. Coenzymes are typically still technically proteins, whereas vitamins are not. I'll get back to you on it. Let me look that up and make sure. Okay.
That makes sense.
Yeah.
I was just thinking like in general.
Yeah.
In general.
But they have coenzymes and cofactors have the same function.
Oh, yeah.
All right. With all that being said, now we've talked a little bit about nutrients. How do they get some of those nutrients in? Well, if this is my cell, I have nutrients on the outside of my cell. They have to bring those in some way. And the process of bringing things in is called transport. Okay, but typically a part of bringing nutrients in is also secretion. So we're going to talk about both secretion and transport and their role in bacterial nutrition. So let me go through an example of what I mean by this. So I have my cell here. again, here's my little cell. Many kinds of nutrients out in the environment. Many of the best nutrients are in the forms of, say, macromolecules floating around outside. So I might have a macromolecule, a protein up here. I might have a polysaccharide up. Macromolecules are very energy-rich molecules, very good source of energy, but also structural building blocks for a cell. So these are nice, good nutrient sources. The problem is they're too big. An entire polysaccharide chain is too big to bring into the cell across the membrane easily. An entire intact protein is simply too big to easily be brought into cells. So how do cells get around that? How do bacteria get around that? Well, what they do is they secrete out enzymes. So if you have these big polysaccharides out there, these big proteins out there, what they do is they make proteases. Proteases are enzymes that digest proteins. They make nucleases. These are enzymes that digest nucleic acids. Amylases, enzymes that digest sugars. Lipases, enzymes that digest lipids. They make these enzymes and then they secrete them out to their surroundings as a form of extracellular digestion. And then if they're digesting this protein, what do you think they're digesting it down to? If you're digesting a protein apart, what are you probably breaking it apart into?
Amino acids.
Amino acids. The proteins are just many amino acids put together. If I'm digesting a polysaccharide sugar, what am I probably digesting it down to?
Glucose.
Could be glucose as an example of a monosaccharide sugar. It depends on what the polysaccharide is. But you're probably taking that polysaccharide and breaking it up into individual monosaccharides then. And that's what's really going to be happening here is they secrete out these digestive enzymes, and then they basically, again, are breaking down that protein into things like amino acids. They're breaking down my polysaccharide, and now they're breaking it down into monosaccharides, for instance. Those are now small enough to transport into the cell. So when I said bacterial nutrition usually involves both secretion and then transport of the digested products into the cell. And this was, again, called extracellular digestion, or sometimes it's referred to as nutrient mining. The other thing to keep in mind is that, again, proteins, macromolecules, those are good energy sources. Good sources of structural building blocks. Guess what's an even better energy source? Other cells. Pathogenesis, those types of organisms that are pathogens and cause disease. There's a great deal of carbon and energy that's sequestered and tied up in living cells, in living things. So you have a pathogen, its nutritional strategy is to create out really strong, toxic digestive enzymes. Things that are strong enough, they tear apart entire cells like a host cell. And they just break it open and eat it, in essence. They take that host cell, they digest it apart, and then they eat it as a nutrient source. And when pathogens get in our body, that's in essence what they're doing. It's a nutritional strategy as far as the pathogen is involved. It's a way for them to steal nutrients from other cellular nutrient sources, in essence. Then, questions about that? Yeah, Eric.
I don't know if this is gonna get answered later, but to me, it seems like secreting all of these enzymes is not very energy friendly.
We will get to that here in a little bit, actually.
Is am I correct in assuming that because of what we're making or what the bacterial cell is making is greater than the loss of the enzyme?
Yeah, as long as these enzymes are hitting a target molecule, they're going to get back more than it costs them to make the enzymes.
Okay.
Yeah. But you're right that it can be a little bit wasteful, and we're going to talk about that.
Okay.
All right, so what we're going to be talking about today and then on Monday is secretion and transport. I'm going to talk about secretion first. That's because secretion does come first when we're talking about bacterial nutrition. They make out and secrete out these digestive enzymes, and then you transport the digested nutrient products in. So we're going to talk about secretion first, but then we're going to talk a little bit about transport as well. Now, different kinds of bacteria have different secretion systems. I'm going to give you through some examples. We're going to talk about sec-dependent, or what's called generalized secretion. We're going to talk about type 1 specialized secretion, type 3 needle complex secretion, twin arginine translocation secretion. These 4 are the ones I'm going to go through in detail, but they are some of the more common ones. There's a whole bunch others, such as type 2, type 4, type 5, type 6, that we're not going to cover. And it's just a matter of these are a little less common, that there are variations of some of these, and that we just don't have enough time. Okay, but I'm going to cover some step-by-step how they do some of their protein secretion. I'm going to start off with sec-dependent, or what's also then sometimes referred to as generalized secretion. All right, the general secretion pathway, or the sec-dependent pathway, those are the same thing. It's called the sec-dependent pathway because it depends on a certain set of proteins called these sec proteins. This is actually, when we're looking at this, the proteins that are labeled here, when I say sec Y, sec E, sec G, this is actually a sec Y protein, a sec E protein, and then a sec G protein. But they form this YEG complex. So sec Y plus sec E plus sec G will then form this integral membrane protein complex that will form a pore or a hole in the plasma membrane of then a bacterial cell. But it's a controlled gate. It's a controlled pore. It's not an open one. It only lets certain things through. By the way, it's always in the plasma membrane. So these sec proteins are always localized to the plasma membrane. Okay. Sec A is another protein that's going to be involved. These are integral membrane proteins. Sec A is a peripheral membrane protein located on the inside or cytoplasmic face of the plasma membrane. Sec A is a peripheral membrane protein that provides the power that will export a protein then. And I'm going to go through a step-by-step what it does, but this particular system secretes a wide variety of proteins across the plasma membrane to either the environment or to the periplasmic space. Why do I say either the environment or the periplasmic space?
Because of the
Yeah. It would be to the environment if we're talking about a gram-positive organism. It would be to the periplasmic space if we're talking about then a gram-negative organism. Because again, this thing is always localized to the plasma membrane. Okay. It secretes a wide variety of proteins across the membrane, but not any protein across the membrane. Secreted proteins have to have the right, what is called signal sequence. This is a structural motif that the secretion machinery recognizes and then allows that protein to get secreted or exported. The signal sequence for the sec-dependent protein is a particular N-terminal hydrophobic alpha helix that is on the protein that's going to get secreted. So it has to have this N-terminal hydrophobic alpha helix, or it cannot be secreted through this complex. So it has to have that particular marker or flag, in essence, this molecular cue. So that signal sequence is going to enter into the sec-YEG secretion system. So it goes through. If it's present. Once it's docked, that kind of opens up the pore. Sec A is going to use ATP hydrolysis as a source of free energy. So the energy source for this one that does secretion is ATP. And that ATP energy source is then used to pump the protein out of the cell. So it goes through the pore complex, but that N-terminal alpha helix immediately also is inserted into the membrane. So it goes out, but then it gets inserted into the membrane right next to the pore complex initially. Okay. And sometimes that's all that happens. Sometimes that's the end. During the secretion, the protein is pumped out in an unfolded state. It's in its primary structure, but once it gets out there, it folds into its tertiary structures. So this is just supposed to be my final 3D shape or tertiary structure. Whenever I was showing the protein like this was supposed to be its primary structure, then unfolded. Once it gets outside of there, it folds up into its tertiary shape. Questions about that? The other thing is it attached to the membrane? My protein that got secreted, is it attached to the membrane? It is still. In some cases, that's it. Some proteins get cleaved by another enzyme called signal peptidase, and then they get released. They'll go off and float into the environment. And that signal peptidase, it recognizes a very specific sequence of amino acids and cuts there. So if they have the signal peptidase signal, they get cut off. Other proteins, though, they may actually remain in the membrane and just function at the cellular surface. So it completely depends on, do they have that signal peptidase sequence or not? If they have it, they get cut and they float off away. If they don't, they stay membrane inserted. And in fact, they may even insert to the membrane a little bit more in the end. Questions then about that?
What's the purpose of that?
So it's going to be secreting out. This might be a digestive enzyme that's going to go off and look for a nutrient. It's going to float off and digest some type of nutrient source out in the environment. This might be, though, a membrane protein. Maybe it functions and does something. Maybe it's like a detoxification enzyme. So if something comes along that might hit the cell and damage it, it actually destroys that and gets rid of it and helps protect the cell. You might want those types of enzymes to stay pretty close to the cell, making sure it's protecting it properly. It depends on the function. All right, one last piece of this. Chaperones. Chaperone proteins. Anybody ever heard of chaperone proteins before? Okay, chaperone proteins are proteins that control the folding of other proteins. Sometimes there's a lot of different kinds of chaperone proteins. Some chaperone proteins just say take a cytoplasmic protein and help it fold up into its right tertiary structure. So help it fold up into its right 3D shape. And that's the job of that chaperone protein. There's other chaperone proteins that actually turns out their job is to keep a protein from folding. So some other chaperone proteins, their job is to say don't fold. And for instance, SEC-B is then a protein that's involved, is a chaperone protein that's involved in the SEC system. SEC-B is here. If a protein is going to get secreted, it has that N-terminal hydrophobic alpha helix. Turns out SEC-B recognizes that, binds to that, and does not let the rest of the protein fold up. It keeps the protein in an unfolded state. But then it also delivers the unfolded protein to the SEC-dependent pathway, to that pore complex. Helps load it onto it. Then when it's loaded, it comes off, and that protein is going to get exported. It's going to get secreted out of the cell. Questions about that? If you didn't have the chaperone protein, this might just end up folding in the cell before it would complex with this, and then it couldn't get secreted. Once it's folded, it's too big to fit through. You guys might have noticed that I had my little shapes here. This is basically bigger than the pore complex. Can't fit through it if it's already folded. Does that make sense, everybody? No questions about that? All right, questions about that sec-dependent or generalized secretion pathway? I'll stop there for today. We'll talk more about different types of secretion systems on Monday, and we'll start going also through transport on Monday then as well. And yes, by the way, if I have stuff that's color coded, that's always a hint that yes, I do expect you guys to remember the names of some of these proteins.